Okay, so next we're going to talk about gene expression, which is composed of two major processes. So that's transcription and translation. So in general, transcription is the process of taking DNA and converting it into mRNA, and translation is the process of taking that mRNA and converting it into a protein. And we'll come back to this. So transcription, again, this is our first step, and during this process, DNA is going to serve as a template to create our messenger RNA or our mRNA. The cell needs a copy of the genetic instructions to send out into the cytoplasm because that's the location of the ribosomes, and DNA cannot leave the nucleus. So in order to get some instructions that are in the nucleus to the site of protein synthesis, which are the ribosomes in the cytoplasm, we need this mRNA in order to kind of carry it there. So how does this work? A specific gene is going to be activated, and an enzyme called RNA polymerase is going to bind to it, causing that portion of DNA to unwind and unzip. So they're showing you the enzyme over here. I want to go ahead and point out we talked about another polymerase in DNA replication. That was DNA polymerase. So if we're taking DNA and we're trying to replicate it to make more DNA, that's DNA polymerase. If we have DNA and we're trying to make it into RNA, that's an RNA polymerase. So that's why we're talking about an RNA polymerase here. So again, it's going to come in, it's going to bind to a specific gene and cause that portion of DNA to unwind and unzip. And as RNA polymerase moves along the DNA strand, it's going to build a complementary RNA strand by matching RNA nucleotides to those DNA bases. It's going to replace thymine with uracil in the RNA. And the result is something that we call a pre-mRNA. And this is going to carry the coded message. So we'll come back to the pre-mRNA, but I'm going to go back up to the genetic code. So the language of the genetic code is read in triplets, which are basically just groups of 3 nucleotide bases. In DNA, these triplets are just called triplets, but in mRNA, we refer to them as codons, and each codon codes for 1 amino acid. The genetic code also includes specific codons that signal where the protein should start. There is one in particular, and that's AUG. This codon codes for the amino acid methionine, which is abbreviated MET. And it also includes specific codons where it should stop. So there's actually 3 stop codons, UAA, UAG, and UGA. So when the ribosome hits any one of these 3 nucleotide triplets or codons, then that's a signal for the ribosome to disassociate, fall off, and basically translation is over. So these codons are guiding the ribosome during translation to make sure that we build the correct protein. So let me show you how this is done. And here's a good example up here. So here's our mRNA right here that we just made. This triplet right here is GGU. So this is a codon. So if we go down to the genetic code, we look for GGU. We see that GGU codes for glycine. So our tRNA molecule, our transfer RNA molecule, is going to come in carrying a glycine, which you can see here, and drop it down here. So the mRNA, again, is just basically the message of the DNA, so we know what amino acids to lay down. So the tRNA is going to match what's called an anticodon to the codon on the mRNA, so make sure it lays down the correct amino acid. Here's our mRNA strand. So the next triplet or codon is AGA. So if we go A to G to A, here's our triplet right there, AGA. It codes for the amino acid arginine. So here's our little tRNA molecule, which is this little teal loop. It's carrying an arginine, and it's going to match its anticodon, which is right here, to this codon on the mRNA via complementary base pairing. So it's going to lay down an arginine next. And then so on and so forth. We can look at the next triplet on mRNA, go down to our genetic code, and look for I forgot what it was, ACC, that's threonine. And if we look at this tRNA that's matching it, it's carrying that amino acid. It's going to use its anticodon to complementary base pair, drop this amino acid off, and we're starting to link together amino acids and build our polypeptide or our protein. You are not expected to memorize this. If needed on an exam, it will be provided to you. I don't even have it memorized. Probably very few people in the world have this actually memorized. But you should understand how to use it. Okay, so basically what we talked about is RNA polymerase coming in, making a copy in the form of RNA. We took our, what should be thymine, and replaced thymine in our complementary base pairing with uracil, because RNA does not contain any thymine. It has uracil. So what we formed at the end of transcription was something called pre-mRNA, if you remember. But before mRNA can leave the nucleus and be used for translation, this pre-mRNA has to be processed. And this processing includes the addition of something called a 5' cap and a poly A tail, whose purpose is to protect the mRNA and help it exit the nucleus. So another thing that the cell is going to do in order to process this pre-mRNA is to remove non-coding segments called introns. So not every nucleotide in RNA will actually code for an amino acid for a functional protein. So we have to get rid of those non-coding segments, which again are called introns. And what we're going to leave behind once we remove the introns are something called exons, which are our coding regions. In some cases, the cell can go in and rearrange or select different combinations of exons through a process called alternative splicing. And this process really just allows one gene to code for multiple proteins. Once the processing is complete, the mature mRNA is going to leave the nucleus and travel to a ribosome in the cytoplasm, where we can begin translation. So here's our mRNA. They're showing you a 5' cap and a poly A tail. A poly A tail is just that. It's a strand of adenine nucleotides that are added to the end, again, to help protect the mRNA. So now we move on to translation, and this is the second major step of gene expression, and this is where the mRNA's message, once it's processed, is used to build a protein. And this process occurs at the ribosome. And if you remember, the ribosome is made up of ribosomal RNA or rRNA and some proteins. So during translation, our transfer RNA or tRNA molecule is going to bring in the appropriate amino acid to the ribosome. I just showed you how that's done previously, where each tRNA has its anticodon, which is a set of 3 bases that are complementary to an mRNA codon. When the anticodon on the tRNA matches up with the codon on the mRNA, then the correct amino acid is added to the growing chain. So that's kind of what we talked about. So translation takes place in 3 phases. We have initiation, elongation, and termination. So during initiation, the mRNA is actually going to bind to the small subunit of a ribosome. A ribosome has a small and a large subunit. So mRNA binds the small subunit of the ribosome. And then a special tRNA that's carrying the amino acid methionine is going to bind to the start codon, which was AUG that I pointed out on the genetic code. After we have that happen, the large ribosomal subunit is going to join to help complete the setup. The next step is elongation. The ribosome, again, has 2 main sites. It has a P site and an A site. The P site holds the growing polypeptide chain, and the A site is where the next tRNA is going to arrive with its amino acid. So once we have two amino acids that are laid down, these amino acids are going to be joined together by a peptide bond, and the ribosome is going to shift along the mRNA, reading the next codon and continuing to build the chain one amino acid at a time. So that's the elongation part. Until we get to the termination phase. And this is when the ribosome is reading along each of the codons, and then it's going to encounter one of those three stop codons. Then we're going to have a protein called a release factor that's going to bind to the stop codon instead of a tRNA, and this is going to cause the ribosome to disassemble. The mRNA is going to be released so that the newly made polypeptide can be formed. Basically, we're releasing the polypeptide. It's going to fold into its final functional protein. So again, gene expression in eukaryotic cells is going to begin with transcription inside the nucleus, where DNA is used to make pre-mRNA. After the mRNA is processed, it's going to exit the nucleus and move to the cytoplasm. There, at the ribosome, is where translation is going to occur, where it's using the mRNA as a guide to link amino acids into a protein with the help of rRNA making up the ribosome and tRNA that's bringing in the amino acids.